There we go. Welcome to this first lesson of the week, for us at least. We are already in week number 9, I believe, right? I'm gonna check. I'm not even sure. I think we're in week number 9. Yeah, we're in week number 9, and this week will be about genomics and biotechnology. It's a very nice week. This one, in fact, it's a lot of content what we're going to do this week, but it's very interesting. It's a pity, in fact, that we only have 1 week to do this, so we can only touch it in a superficial way, because there is so much to tell about this genomics and biotechnology that we could spend the whole course on it. In fact, it's one of my favorite parts because I study this, so this is one of my specialties. So this week it will be about DNA and genomics. So before we start, I just want to share with you what is happening tomorrow. This don't forget, we have this change maker day. This is not something that we're going to do in the classroom, but I asked you to get registered so you can have a look at it. This is not something of our course, but I think it's important that you guys get invited in this kind of events because they're very interesting. So here you have the agenda. If you can have a look, it starts at 9:30. So maybe if you're free at 11, you can participate in the Consciéntate con un turismo consciente. They're very nice talk. Consumo local, consume responsable. This one you can also follow. They're very interesting topics. I don't know if you can ask another teacher to perhaps touch one of the subjects, because they're very interesting things here. Also, it is until 8 in the evening. So you have here the whole afternoon. You can take talleres, very interesting for you to go. Okay, so there's compartiendo herramientas y experiencias de cambio, perspectiva de género. These are all things that should be close to your heart, and I really think it would be nice for you to assist to certain things. Okay. So here you can have a look at it. I just share it with you. We are not coming back to this in the classroom, but it's open for you, so you can participate with this. Okay. So like I said, what are we going to do today? Today we're going to give an introduction of DNA. So last week we were in the plasma membrane. Yeah, remember that? We were investigating and having a close look how the plasma membrane was formed, what its function was, what kind of proteins has it, how transport, one of the important functions of the plasma membrane is transport. And now we move into the nucleus. Okay, we're inside the nucleus now. Remember that every cell, every eukaryotic cell has a nucleus. Prokaryotic cells not, but we're going to focus on the DNA of eukaryotic cells because it's way more complex, the eukaryotic DNA. So we're inside the nucleus, and we're going to see how this DNA is inside the nucleus, how out of what does it consist, how did they discover it, what is its function, and how does it work basically, because it's very special what's happening in the nucleus. So let me share with you what we're going to talk about. So here you can see we're gonna start sharing. So what you see, can you see the model? So this is how a DNA molecule looks like. Okay, I'm sure you have seen it before. It's been the logo of many companies. It's a very iconic thing, a DNA molecule. You know it like this twisted stairs. It looks like that. It's called a double helix, and that's what we're going to have a look at. Before we start, I'm going to show you in a moment a video. about DNA, not about DNA, but it's more like a video about your DNA and what its function about how we are all related are. It's a very nice video. But before that, I want to give you some mind-boggling numbers. You don't have to remember this, but to give you an idea what we're talking about. So who has an idea how many cells do we have in our body, more or less? We don't know exactly, but we have a good estimation, which is very close to reality, because we measure like a little part and then we multiply it, and we have a good idea of how many cells a human body has. Of course, it depends if the person is small or tall, a bit more bigger, let's say, yeah, or thin, you know. So how much do you think? How much do you think that is? Just give an estimation.
1 trillion.
No.
I'm gonna say 10 billion.
No. It's
Is
more
it more
in
or
the...
less?
What, mister? I didn't hear.
How many cells does a human body have? Okay, we're
100
about
billion.
7-8 billion people on planet Earth. Okay,
Like a quadrillion?
yeah, it's more than a trillion. In fact, it's 37 trillion cells. 37 trillion. That is a lot of cells, you know. I mean, that is 1,000 times more, or it's way more than there is people on planet Earth. It's a lot of cells. Okay, so every cell except red blood cells of those 37 trillion cells has a nucleus. Okay, inside that nucleus you have a complete DNA strand. Okay, this DNA is enough to make another human being. It has all the information in each of your 37 trillion cells. So how long is the DNA in that mini tiny nucleus that you have in the center of the cell? How long is the DNA, the human DNA in the nucleus? How long is that? Just
Until the
estimate.
cells die. On.
Enough to go to the moon and back.
No, it's not that long.
Like 10 seconds.
No, how long? Let's think that the DNA is a thread. You take one cell and you pull the DNA out of the nucleus of one cell. How long would that DNA be?
Meter.
A meter, yeah, more or less. It's 2 meters long, more or less, in each of those 37 trillion cells. Now,
like, mister,
yes,
like something like really small, it can get like 2 meters.
yes, exactly. It's so thin you could not even see it with the naked eye. It's so thin, the DNA thread. So now imagine that you take the DNA out of each of the nucleus of each of your cells and you would connect it. Now, this is going to be mind-boggling how long that will be. How long do you think that is? If you have 37 trillion cells and you have in each cell 2 meters of DNA, how long is it going to be?
Wait, I can
that,
search
no?
it. I can use a calculator.
Probably bigger than the solar system, no? Something like
Very
something
good,
ridiculous
Santiago.
like that.
It would be a length of 278 billion kilometers, or twice the solar system, or it would be the length of 1.5 light year.
Like 740 trillion meters, like something like that.
Yeah. So this is very remarkable, guys. Think about that, how complex life is with all the DNA we have inside us, how incredible that is. And it's not only the numbers that are incredible, it's also what DNA does. And therefore, first we're going to have a look at a little video, which is nice because we are multicultural, and there is so much racism and so much confusion and problems in the world. And it's a very nice video showing us that in fact, even you and me, I'm from Europe, but we could be even more similar than we think. Just watch at this. I'm going to share the video.
I'm proud to be English. My family have served and we've defended this country and we've been to war for this country.
I'm really patriotic about Bangladesh.
Well, I am 100% Icelandic, yeah, definitely.
This is a Kurdish wedding with my mom in the traditional Kurdish clothes.
We're just proud black, so that's it.
Yeah, I think we are probably the best country in the world, if I'm honest.
Think about other countries and other nationalities in the world. Are there any that you don't feel you get on with well or you won't like particularly?
Germany. Yeah, I'm not a fan of the Germans.
You might think they're a little bit...
Particularly India and Pakistan, probably, because of the whole, the conflict.
Because I have this side of me that's like, that hates Turkish people. Not people, but the government.
But French?
No. We're just best, it's just fact.
I'm more important than you. I don't know you, but in my opinion, I am strong and I am more important than a lot of people.
How would you feel about taking a journey based on your DNA?
Yeah, I feel very intrigued.
What could you possibly tell me that I don't know?
So do you know how DNA works? So you get half from mum and half from dad, so 50% from each of them, and they get 50% from their parents, and back and back and back. And all those little bits of your ancestor, they filter down to make you. I need you to spit in this tube for me. You spit up to the little black line.
That's a lot of spit.
Right, the story of you is in that tube. What's it going to tell me?
It's going to be, oh yeah, you're French, and wait, your grandparents are French, and wait.
100% Bengali.
Solid Iraqi.
I'm Cuban.
You're going to tell me that I'm English? Like I've told you.
Jay, can you come down and join us?
I'm a little bit nervous, I have to say.
So you ready to find out your results? Will you read it out to us, please?
Wow, look at me. Oh my God. Oh, wow. Shit, I didn't expect that.
All of them.
Whoa! No.
Caucasus, which was Turkish?
Yeah.
Eastern Europe, Spain, Portugal, Italy, Greece.
I'm 32% British.
What? Great Britain, 30%. Can we 5% German?
I'm Irish.
Yeah.
So I'm a Muslim Jew.
Great Britain, 11%. Are you sure these results are mine?
Eastern Europe?
Iceland has definitely moved closer to Europe now.
I'm going to go a bit far right now, but this should be compulsory. There would be no such thing as extremism in the world if people knew their heritage like that. Who would be stupid enough to think of such thing as a pure race?
In a way, we're all kind of cousins, in a broad sense. In a much more direct sense, you have a cousin in this room. Turn around and guess who it is. Waj?
Yeah. What's that?
Why don't you come down here and meet your cousin?
God, this is a
Did you know that?
I did no idea. This is my heart's pounding right now. I swear to God. We met. I'm Jay from everywhere, but I read to this.
I'm a real man of the world.
Wow, that's beautiful. Thank you.
So would you like to travel to all of these places?
Yeah.
Oh yeah.
So guys, isn't that beautiful? In
Yes, sir.
fact, yes.
Tomorrow I'm going to do the DNA test. Nah, I'm joking, but I would
Oh,
like to.
in fact, we should. I mean, I'm very curious about you guys because I mean, Mexico is so special in so many ways. Sorry, it's the sorry. Sorry, yeah, it played another video. What I want, what I think is so special about you guys is that look, if I do the test, what they do now, I know what will come up. I guess it will be like I'm a little bit German and I'm a little bit Dutch. I'm a little bit from Sweden. It will be Europe, but you guys, you're way more interesting because many of you, if we would do this to you, you would also be linked to Europe because you're not indigenous Mexicans. None of you is. You might have some indigenous blood, some of you, but most of you have European roots, if you like it or not. And that's what makes you so interesting. And I would love to do this with you guys. Of course, I cannot do that, but it would be very interesting to see because especially United States, Mexico, Brazil, these kind of countries, the population is not you're not the original population of Mexico. I mean, look at yourself and so many of you how white you are. Okay, you don't look like um, you don't look like Mayan people in the south or women from Chiapas. You look more European, you know. Let's be honest, and that's why I think it's really interesting to know how much Spanish are you, how much Portuguese, how much French are you, because you all are. Even if you don't think so, you all are, because you're all a mix from European people that colonized Mexico and
Yes, sir.
and some people that were still left behind, like the indigenous ones. Yes.
I know like the four countries that I came from. Like I have Chinese descendants, well, genes, Arabic, French, and American.
Mhm.
I think.
It's very interesting, this, Emilio, because I don't know if you know that, but have you ever did you ever travel to Chiapas or the Tarahumara or something? Have you noticed that indigenous Mexican people, they look sometimes a little bit Asian? It's because they are more they're more related to Asian people than they are to European people, because the people that came to the Americas, they crossed the Bering Strait and they came from Asia. Anyway, what I want to tell you is that maybe you think like I'm the foreigner, but maybe we are more related than you think, you know. Maybe we have the same ancestors, you and me. So I think that's very interesting. So let's have a look to this DNA, guys. First and for all, there is a lot of information. I first want to ask you, don't get scared, because you don't have to know all the details, okay? What you have to know is basically how the DNA molecule is built, what it does, and a little bit of the history, and the two big processes is how, like you can see in my background, you see a double DNA helix, which is transcribed to RNA molecule, and then it will be translated to a protein. So we will have a look at that later, okay? Let's first have a look at the DNA itself and little history, okay? You just have to know the main idea of the history, but it's interesting to know where it comes from. So let's have a look. The first person that we're gonna introduce is Friedrich Mischer, okay? Friedrich Mischer. This was already in 1869, and what, so it's 150 years ago almost. So what this guy did is that he was the first one that noticed there was some kind of acidic substance inside the nucleus, okay? He would call that substance DNA. He had no idea what DNA was. He just defined it for its chemical purposes, but he had no idea that DNA did what it does, you know, that it has genes and that it is the code of life. He had no idea. He just identified it as acidic substance inside the nucleus. In fact, in those days, they had little idea about genetics whatsoever. And whatever characteristic went from one person to another, they would think it was proteins that carried the information. So in the 1920s, Griffith, he came with the transformation principle. So what is the transformation principle? In fact, it's a thing that that is a proof that certain traits can be passed on. Okay. So what did he do was basically he would inject a mouse with bacteria. Okay, and then he would see that the mouse would live. Okay. Whenever he did like bacteria that would kill the mouse and he injected, then the mouse would die. So you have like blue bacteria that doesn't cause harm, the mouse lives. The bad bacteria, you inject the mouse with the bacteria, the mouse dies. Okay. So what did he do now was, if you kill the bacteria, the red bacteria, and you inject the mouse lives. Okay. But if you combine both of them and you inject them, the mouse dies. So this was the transformation principle, because basically what happened is that even if you kill off the bacteria, if you combine it with a living bacteria, that this living bacteria could absorb something from the harmful bacteria and injected, the mice dies. So the transformation principle implies that there is some kind of way that living things can absorb some kind of genetic threat and causing the mice to die. Is this clear? Because it's a little bit complex, but you don't have to know the details about it. But Griffith basically was the first person that indicated that that there is something inside cells that can be passed on to next generations, even if you kill it off. There must be some kind of molecule, or there must be sometimes something inside the cell that has a certain characteristic that when you inject it, that this with living cells, that the characteristic stays alive. Okay, this is called the transformation.
So, Mr.
Yes.
So it's like some scientists said that some viruses and bacteria will evolve like to an evolution, and they will be immune to vaccines. So is the same with the mouse example, or?
Yeah, it's similar in fact,
different?
because what you have here, it's non-virulent. And what non-virulent means, it's a bacteria that doesn't cause harm. Virulent is a bacteria that does cause harm. If you kill the harmful bacteria with heat, okay, then the mice would live. But what happens if you combine them is that the healthy bacteria would absorb some genetic material, but they didn't know what the genetic material was. They just observed, you know, and this is called the transformation principle, that this is an experiment that has been done that they couldn't explain. They didn't know which kind of molecule is causing the effect, which is that the mouse dies, you know. That's the idea. It's an experiment that they became aware of, and indeed it's very close what we do with vaccination. In fact, you know, this is basically vaccination, what you do. Okay, so that's basically Griffith. Then we go to Oswald Avery and MacLeod and McCarthy. There were three persons, and what these persons did is, you see here a list. You don't have to know that list, but basically, to make a long story short, is that they would they build it on what Griffith did. Okay, they wanted to know what molecule is responsible for the effect we see in mice. So what they did is they would add enzymes to the mix, and they would eliminate all the time one molecule. They would eliminate RNA. Okay, the effect is still there. It's not RNA. They would eliminate the DNA with a DNase, which is the enzyme that destroys DNA, and they would see that the effect disappears. So they were, in fact, the persons that identified the molecule that is responsible for the changes that you saw in Griffith's experiment. So which molecule is responsible for the effect you saw in the experiment of the transformation principle. Okay, so in fact, they determined that neither protein nor DNA is genetic material. So that left them with still protein and DNA. So they eliminated a little bit the options, because it's what is causing the genetic, what is causing like the effects could be DNA, could be RNA, could be a protein, or could be other molecules, and they eliminated the possibility it being RNA and being proteins. Okay, so then in the 50s they did another experiment that confirmed the DNA's function. Okay, so DNA was getting more and more a candidate to be the carrier of genetic information, and with this experiment they had even more strong evidence that indeed DNA was the best candidate for it. What they did was, in fact, there is viruses which we call bacteriophage, and a bacteriophage is a virus that can infect a bacteria. Okay, so what happens basically, they did an experiment and they discovered a bacteriophage, a virus that infects a bacteria. And with this experiment, they showed that they could make certain bacteria do things that they normally would not do. Okay, so it's the bacteriophage that injected a little bit of DNA in the bacteria, and the bacteria would do a certain function. Did you know that nowadays, what you see here, what was already discovered in the 1950s, this is now a very important thing in biotechnology. What they do is they have a bacteriophage, and they put a little piece of DNA in the bacteriophage, and they let it inject bacteria. And for instance, the DNA that they inject in the bacteria is a DNA that, for instance, helps them eat plastics. And that's how they are making bacteria that can biodegrade plastics. You know, they don't exist in the natural world, but we are manipulating those bacteria in order to do them whatever we want. Did you know that we even have bacteria nowadays, also with the help of bacteriophages, that we let them make certain kind of gasoline? There is bacteria that can produce gasolines, or they can produce insulin, you know? Did you know that all diabetes persons in the world, they get insulin made out of bacteria, and those bacteria, they have in fact DNA, and we just put a human gene into the bacteria, and the bacteria is basically producing human insulin. So this is a very important characteristic, and it was the proof in the 1950s that the DNA is indeed the genetic code of life, in fact. Okay. So in the 50s, like we see here, thanks to the previous experiment of Hershey and Chase, they accepted that DNA was the genetic material for all living organisms. But there was still one big problem. They knew of which molecules the DNA was built, which is adenine, guanine, thymine, and cytosine. Here you see the chemical formula. You don't have to know that. But they had no idea how the structure was. They still didn't know that it was like a helix all twisted. They had no idea how it was built. They just knew that you had adenine, guanine, thymine, and cytosine. And the thymine and the cytosine, they call them pyrimidine bases, and the adenine and the guanine purine bases, because this have a double ring and this has a single ring. Okay, purine a double and pyrimidine a double one. And what they also noticed is that in 1952 they noticed that the amounts of those bases, you know, the purine and the pyrimidine, they had like the same, how to say it, they had they had a similar percentages, you know. You would have the same amount of thymine and adenine, which was 40%, and the amounts of cytosine and guanine would be 60%. And this led them to the conclusion that A always combined with T and G always combined with C. Okay, so adenine always combined with thymine, and guanine would always combine with cytosine. Guys, I know this is a little bit complex and we go a bit fast, but what you see here, this A, G, T, and C, This is in fact the genetic code, okay? These are molecules and they represent the genetic code. And how you can remember this very easily is, who can tell me how a computer works? Who knows
With
that?
a binary code with
Yeah.
ones and zeros.
Exactly. A computer works with a binary code. So a computer is 0011011. It's a code. Basically, DNA does exactly the same thing, but instead of ones and zeros, it uses A, G, T, and C. And if you look to a genetic code, you would see A, A, T, C, G, 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 T, I, C, He, P, I. All those letters, they are in a certain sequence, and that's the genetic code. It's thanks to these molecules that that the DNA strand is made. Okay.
So, Mr.
Yes.
AG and TC, AG always work together, making the 40%.
No, we see that in a moment. A always combines with T and G always combines with C. But basically, you have a double strand, so they combine with the other strand. But basically, the code itself, it can be any combination, okay? So here you can see different kind of organisms, and you can see here the proportion of adenine, yeah, the proportion of guanine, the proportion of cytosine, and the proportion of thymine. And what you indeed see is that the G and the C, they have sort of the same proportion as the A and the T, indicating them that they combine, okay? And then Franklin is the other person, and is the first person, in fact, that made this picture. It looks a silly picture, but it's X-ray X-ray diffraction she did. And basically what you see here on the bottom, this X, it was the first picture of DNA ever taken. And thanks to this picture, it was Watson and Crick in 1953 that really came out with the double helix model, what you see here, okay? So it's those two persons that made the structure of the DNA helix, okay? So they said that how are the atoms and the molecules combined and how do they form this model? There is an interesting documentary about this that I can share with you if you can watch it. It's very nice. And they came to these conclusions, okay? So that's the history. I went a bit fast, I know, but it's not that important, you know. It's just that you know how DNA came to be and which steps did it take in order to come to the idea of the DNA molecule, because they had no idea which molecule was containing genetic material, what was it made of, and how the molecule did look like. It took them more than 60 years, in fact, to come to this model, okay? Okay, so everything clear so far, or you want me to go back over the history, something else you want to, because now we go a little bit more to the content of the molecule?
I want to see the names again of AT and GC, please.
Here you have them. So this is the adenine molecule, the guanine molecule, the thymine molecule, and the cytosine molecule. Okay.
Okay. Yes.
And basically, the human DNA has a sequence of those letters, which is really, really long, and they form genes. But we're going to see that in a moment in more detail. Okay. What you basically have to know now is that the DNA is a double helix. Okay. And that the helix is made of those base pairs, we call that. A connects to T and G connects to C. And that they are held together with a kind of force that we call hydrogen bonds. So here you see hydrogen bonds. This is the force keeping the two strands together, like you can see here. But we're going to see that in more detail in a moment. Okay.
Sure. I have a question.
Yes, go ahead.
Does DNA controls like the way the cell is formed, or no?
What do you mean the way the cell is formed?
like if the genetic material, the DNA, controls how is structured the cell, or no?
Indirectly, yes, but basically, whenever you have a new cell, we have seen in the cell theory that cells come from other cells. In fact, before a cell comes into existence, what happens is that the DNA is the first thing that is copied. So it's one of the first things that a cell has before it becomes a cell, it's the DNA. So indirectly, yes, but basically the cell becomes more the cell what it has to be because of the surroundings. There is like chemical messengers that tell the cell, for instance, my skin is growing and my skin cell is duplicating, but this cell is pre-programmed to become a skin cell, but it's also thanks to the surrounding cells that send messages, chemical messengers that say, oh, please convert into a skin cell. So basically, it's a very complex mix of DNA, of chemical messengers, and of the pre-programmed status of the cell. It's very, very difficult, this, Emilio, the details about this. It's a very specific question. To go back to this, like I said, DNA is the first one that is produced. So that's why we call it DNA replication. It's copying the DNA molecule. So whenever we're making cells all the time in our body, and the DNA replication is in fact the first thing that happens. And what happens is that the DNA strand, like you can see here, C combines with G and A combines with T. Whenever we make a copy, the DNA strand goes open, like you can see here, and that's what we call a replication fork. And our cell, inside the cell, we have these molecules that are loose, and they come inside the nucleus and they combine with the open DNA strand. And that's how we can make perfect copies of the DNA, because you see here that on the top that A is combined with T, but when it's open, A will always combine with the new T. So this T will combine with the A, this T will combine with the A, this A will combine with the T, and that's how you get a perfect copy, because those molecules, they are like key and lock. A can only connect with T and G can only connect with C. So whenever the DNA goes open, like you can see here, so this is in a schematical way. So here you have the code. This is the code T, G, G, T, A, C, A, G, G, G. So what happens when you copy a cell, the DNA goes open, like you can see here, which is the blue one, is the original one, and it gets filled up automatically with the green one with the complementary strand. You see that? So C will always connect with G, A will always connect with T. So basically, you're making an automatic copy, and it's really beautiful, this of life, because it makes it really, really difficult to make any mistakes, because these molecules can only connect like this. So in the end, what you get is you get the similar strand as the original one, okay? Just because they go open, the molecules, they fill in the gap, because they can only connect with that one, and that's how you get a DNA copy. And this is what we call DNA replication. You don't have to know more than this. It's way more complex, but that's the principle, okay? And here you can see how that works. So what is doing the job? You see here the original parental DNA, yes? And it's like an enzyme, which we call DNA helicase, which opens the DNA. Okay, you see here, it opens the strand, and it's another protein, the DNA polymerase, that fills in the gaps. So you see here, the blue is the original one, and then the green one is constructed, okay? And then here on the other strand, it works reverse, because otherwise it would be a complementary copy, but that's too much detail for us now. Just know that what happens in DNA replication, the DNA helicase opens the molecule, then the DNA polymerase is getting the new nucleotides. Nucleotides is G, C, T, and A in place, and you get a new strand here, and you get a new strand here. And that's what the DNA replication is all about, okay? And these are some proteins that are involved in that process. And what we're talking now, this is happening in the nucleus of a cell when a cell decides to split into two cells, okay? That's the first thing they do, is duplicating the DNA, and the term for that is called mitosis, okay? But we will see that in another chapter, how the mitosis is working. Now we just focus on how the DNA is duplicated, okay? So I go here a bit faster because this, we don't need to know all the details, okay? And then for tomorrow, I'm gonna leave how, if you have the DNA duplicated, we are gonna make protein out of the DNA, because you know that DNA is made, that it is the base for the protein, but that work is what we're gonna see tomorrow, how the protein is built. I think it's enough for today to go until DNA replication. I just wanna know if everything is clear until now, otherwise I give some more details about the certain points. Anyway, we will come back to it. We will review everything again tomorrow. But I just want to know, is everything clear so far?
I got a bit confused after like the protein, like when we started like the replication, I think. Like I got confused.
Okay. So what we saw today is in fact how the DNA molecule is built. So we saw a little bit of history, how they identified the DNA molecule as being the genetic material. And then in the last part, I explained a little bit, the DNA has to be copied, okay? Because whenever a cell splits, you need to make a copy of the DNA. And the copying of the DNA, we call that DNA replication. And that's what I explained. How the DNA is copied is how the twisted DNA molecule opens and it gets automatically filled up with nucleotides that arrive into the nucleus from outside the cell. So they can be produced inside the cell, they can be absorbed by the cell. So these nucleotides, they come inside the cell, they go to the nucleus and they form the new DNA. And before the cell splits into two, in fact, the cell has not 2 meters of DNA, it has 4 meters of DNA because it duplicated all the DNA inside its nucleus. And whenever the replication of the DNA is complete, the cell is ready to split into 2 cells. Is that okay, more clear?
Mister, I have a question.
Yes, go ahead.
Yes, thank you.
Why the AT and the CG like get together? Are they attracted?
That's also a specific question. In fact, it's in the, let me share that quickly with you to give you the reason why that is that they combine. In fact, you have to, you didn't get chemistry yet, but it's in the nature of the molecule. Where is the molecules? Here. So they combine together for a very specific reason. It is because of the hydrogen bonds, okay? So A and T combine because you have here on the edges of the bases, you have a C double bond O, you have here a methyl group, you have other C double bond O, and the forces that exist between these atoms, okay, because there is hydrogen atoms, they can only connect with this molecule, you know, because they have like 3 hydrogen bridges. And if this, for instance, if you have A and G, you see that they're both big and they would repel each other because they take more space. They have more, they have more volume. So they cannot combine together because they have both a double ring. And that's why basically it's the chemical nature that makes them combine. It's like a double ring combines with a single ring, and this one combines with that one. And it has to do with the atoms you have on the ring. Here you have the nitrogen, here you have oxygens, and they create certain forces between each other that can only attract each other. And that's why A will always combine with T and G will always combine with C. It's because of the chemical laws between the atoms of those bases. In fact, hydrogen bridges, there is attracting forces and there is repulsing forces. And the combination of both cause that those molecules are attracted to each other, they fit into each other, and the others, they don't.
Okay.
Yeah.
So the molecule balances it's off with the other molecule.
Exactly. That's very beautifully
Okay.
said. That's exactly what happens. Yes. You're so great,
Teacher.
Emilio. Yes, Anna?
What was like the name of the things that fill up the DNA when it splits up?
These are the nucleotides. So we call them nucleotides or nitrogen containing bases. That's the same. We have two types, the pyrimidines and the purines. But if you say nitrogen containing bases or nucleotides, that's all fine, okay? So it's in fact what you see here in this presentation, nucleotides. Here they call it nucleotides, okay? G is one that come in, but it's all the same thing. So this is the same thing. Nitrogen containing bases is exactly the same as nucleotides, okay? Or bases, if you want, you can call them bases. It depends what kind of book you consult, how they call it. I prefer to call it nucleotides, to be honest, because it's the easiest word and you immediately know what you refer to. However, a lot of books, they call it base pairs because it's a pair of bases. But that's scientists, you know, they can never agree how you call it. And these are some enzymes. Enzymes are proteins. So these are proteins, you can see that, and they have a certain function in the DNA replication, okay? You don't have to know the very much details of this knot. What I would like you to know is that the helicase opens the strand and the polymerase fills it up, okay? And the primase and the ligase, they do gluing and cutting, this kind of things. So we don't have to go in so much detail because we really don't have the time, and moreover, it would confuse you too much, I think. So that's it for today, guys. I hope you enjoyed this class. I know it's a bit complicated in the beginning, but that's why I try to go a bit more slow in the end. So today we have seen how the DNA gets copied, and tomorrow we're going to see how it transforms into RNA, and then from RNA into protein, okay? And then on Friday, we talk a little bit about biotechnology, which is also very interesting. So that was it, guys, and we see each other tomorrow, okay?
Goodbye, have a good day.
Thank you, teacher.